Hello everyone, my name is Martina Paccini and today I will present you a work made in collaboration with Giuseppe Patanè and Michela Spagnuolo about the analysis of 3D segmented anatomical districts through grade levels mapping. In the medical field, especially in radiology environment, there are two main approaches to anatomical structure analysis, which are geometry processing and image processing. These two techniques provide complementary information. Geometry processing focuses on 3D surface model obtained from the segmentation of the original image. These 3D models are useful to perform a morphological analysis of the anatomical structure, extracting shape and geometrical information. At the present state, to our knowledge, color and texture are used only for realistic illustration purposes, because the information provided by the image context are lost in the segmentation process. Image processing instead work on image raw data and permit to extract photometric informations related to the pixel value on the image. In this work, as an example, we use T1 low-field MRIs, where the color of the pixel provides information about the signal received from the tissue. This signal, in turn, depends on the composition of the tissue itself. Mineral-rich tissue will result as black pixel. Collagenous tissue, as well high-bound water tissue, will result dark and intermediate gray levels, while fat tissue, like bone marrow, will result white. The 3D nature of the anatomical structure in this kind of processing is not represented explicitly as happened for the geometry processing. The analysis of anatomical structure in clinical practice is fundamental for different aspects that go from early diagnostics, surgical planning, to follow-up evaluation and therapy planning. In this work, we focused on the carpal district, with a dataset composed by 3D MRIs of the district, the segmentation of these images, and the 3D surfaces extracted from the segmentations. Since geometric and photometric information are both useful, but the two processing techniques run quite parallel, the main question of this work was how is it possible to mix image and geometry effectively? Meaning, is it possible to obtain an approach to represent segmented surfaces augmented with information about their volumetric context? Our idea was to map the gray levels of the image voxel in the segmented surfaces through a volume surface correspondence map, which defines a gray level texture on the surface. In this way, the texture links the image context, that is, the volume in which the structure is inserted, to the 3D geometric representation of the segmented shape. To obtain this integration of information, we needed a data structure able to function as a bridge between the volume and the surface domain. Considering that the volume MRI is represented as a DICOM volume composed by a stack of 2D slices, and that on each slice pixels have a specific dimension, which is called pixel spacing, and an interslice space, which is called thickness, then, to obtain the required data structure, the image volume is loaded into a grid whose elements have dimension x spacing, y spacings, and thickness, with a total number of voxels equal to the product of the number of pixels in each direction. In this way, every grid cell has its 3D coordinates to locate it in space, and each grid cell is associated with a voxel and its gray level. Here on your right, you can see an example of 3D grid, with an orange element that represents a voxel located in the grid by its 3 intervals in the XYZ direction. Moreover, each voxel has its own centroid, described by 3D coordinates. On the other hand, the segmented 3D surfaces are represented as triangle meshes, composed by a set of vertices and triangles. We assume that the mesh is represented as a standard indexed list of 3D vertex coordinates, which describe the geometry, followed by the conductivity, describing the adjacency relations. The two inputs of our method, that are the image volume and the segmented surface, have consistent reference system, since each surface is obtained from the relative image. For this reason, the superimposition of the two doesn't require any registration. Now, to integrate the shape knowledge with the 3D image and include information about the volume around the segmented structure, we explore the gray value in the proximity of the surface. We developed 3 different ways to explore the volume around the surface. In each of these methods, the vertices that compose the surface will be associated with the gray levels of the correspondent voxels in the image. This means that each vertex will be associated with a voxel. What distinguishes the tree exploration method is how the correspondences are identified. Here you can see the list of the method, the Euclidean mapping, if the surface vertex get the gray level of the voxel closed to it, with respect to the Euclidean distance, the internal mapping, where the closest voxel are searched only inside the surface, and the external mapping, where the closest voxel are searched only outside the surface. The internal and external mapping are based on the distance from the normal to the surface on the vertex considered. On the left, there is an example in 2D of the different kind of correspondences, and on the right here you can see a real application where the red is the Euclidean, the green the internal, and the blue the external result of the different correspondences. The result that we obtain is a 3D surface model colored with degree levels of the image. As you can see, the result changes depending on the method used to explore the volume. This is because searching the volume in different extents and directions means to encounter different tissues. The Euclidean mapping texture represents the tissue located in the exact position of the segmented surface. The internal mapping will encounter the tissue inside the bone, that is the bone marrow, which is brighter. The external mapping instead will encounter more collagenous tissue like tendon, ligaments, muscle, etc., and for this reason it results darker. On your right, you can see a graph that shows the mean value of the gray levels for each of the 8 bones of the district. The blue brighter values are related to the internal mapping, the red to the Euclidean, and the darker green to the external mapping. Once we have obtained the integration of volume information on the surface model, studying both texture and geometry information, we can find different applications useful for the clinical practice. Indeed, we can improve those descriptors that previously were related only to shape and geometry. This means providing to medical doctors key information while solving classification, annotation, and landmark identification problems on this 3D enhanced surface. The first application regards the evaluation of the performed segmentation. What we want to know here is which tissue has been included on the bone shape and which not. To do so, we exploit the gradient information between inside and outside the surface. The outer part of the bone is constituted by cortical tissue that results black, while the inner part is constituted by bone marrow and lamellar tissue, which result brighter. This means that if the edge of the bone is placed between the cortical marrow and the bone tissue, then the internal mapping should be brighter than the external mapping, leading to a positive value of the gradient. If the edge is placed outside the cortical tissue, then darker internal mapping ray levels will make the difference negative. Here, the red region of the 3D scaphoid bone represents the location where the cortical bone was included in the bone shape, which correspond to the areas where the internal mapping is darker than the external one. Comparing the segmentation of two bones, it is possible to evaluate which one has a higher portion of cortical tissue included in the bone object. In this cumulative histogram of the absolute difference between internal and external mapping, the hamate bone that corresponds to the blue curve presents a higher portion of cortical bone inside the surface. Indeed, the steeper trend indicates that a higher portion of the bone presents a low difference between inside and outside. The red cumulative histogram, instead, shows a gradual growth, meaning that a larger portion of the segmentation edge has been placed between the two distinct tissues. Another application is the characterization of relevant anatomical regions in order to provide richer and more accurate information to surgeons and physicians. In this case, we integrate previously developed shape descriptors with the image information given by the texture. The shape descriptor that we consider is the shadow map that helps to identify articular region and gives information on the single bone as well as on the district organization. The idea of the descriptor is that the articulation regions are the area of the bone that face to each other, and these areas can be defined as the shadow cast by each bone on the other. Once identified the articulation facets, it is possible to study the gray levels distribution in those regions, and comparing it with the region not involved in the articulation. What results from the analysis of external mapping in this case is that in correspondence of the articulation region, the gray levels of the texture have a wider range, touching also brighter values. This is coherent with the tissue composition of the articulation capsule between two bones. This means that the mapping method brings information not only at a global level, but also at a local one. We performed a one-way ANOVA test to demonstrate the statistically significant difference between articulation and non-articulation regions. For all the bones, the ANOVA test confirmed the significant difference, except for the trapezium and the pisiform. However, the trapezium presents a wider range as expected in the articulation grade level distribution. The pisiform bone instead can easily result as an outlier, since it is the smallest bone of the district and presents just one articulation site. Considering the overall district, the articulation areas actually present different grade levels distribution with respect to the non-articulation regions. Follow-up analysis, especially for degenerative diseases such as rheumatoid arthritis, is fundamental to deduce if the therapy is effective and how the illness is progressing. The idea is to combine shape and texture analysis to find the erosion region developed on the bone of the same patient in different periods of time. To do so, we registered the bone at follow-up time onto the bone at baseline time. Then, we computed the distances distribution between the two bones. In this way, regions of the bone that presented higher distance were considered as possible erosion. Moreover, on the follow-up bone, we mapped the volume information at baseline time and at follow-up time to investigate if a change of texture could confirm the presence of an erosion. It is clear from the results that in correspondence of the erosion site, that is the red region in the distance distribution, there is also a change in the texture. At follow-up time, the external mapping results brighter, accordingly to the tissue changes that are visible in the MRI. Computing the difference between the mapping obtained in the two different times, the higher texture difference results in the same area that was identified by the distance distribution. Moreover, the same coherence is not present in the areas where the high difference distribution values were due to the registration phase. This means that the texture analysis helps in correcting misleading information that arises from the morphological comparison. To confirm what just described, we used the QQ plot between two bone texture distribution to see if the texture changes were significant. On your left, you can see the result of the plot at baseline time. Both bones do not present erosion, and their distribution can be considered equal, since the plot lays on the bisector. In the follow-up, instead, the scaphoid bone presents erosion, and the two distributions can no more be considered equal, as confirmed by the plot deviation from the bisector. In future work, we will focus on the possibility to apply the mapped surface to identify ligament insertion sites. These sites help to classify the pathology at its early stages and to start a patient-specific therapy, so they are relevant for early and specific diagnosis. To identify the insertions on the surface by the mapping result, the range of grade level characterizing the different tissues have to be analyzed. Since in correspondence of the ligament insertion the bone tissue changes into a fibrous composition, the insertions should belong to the brighter range among the dark grey levels. Analyzing the histogram of the external mapping, we tried in this preliminary work to identify the range of grey levels corresponding to the ligament insertion with specific threshold for each patient. Once this range is computed, the vertices that present a ligament insertion will be the ones that are associated to a grey level corresponding to the range. Preliminary results look promising, as you can see in the comparison of a 3D atlas with the potential ligament insertion identified by the red region in our 3D model. Concluding, the proposed method is simple but effective. Its simplicity permits to apply it to different human districts and independently of the surface extraction method. The immediate result of the method is to provide an enhancement of the shape visualization with the volume information. The volume information can be integrated easily in different shape analysis applications, such as segmentation analysis, shape description as seen for the articulation region characterization, shape comparison as in the erosion identification case, and landmark identification such as in the ligament insertion site. Thank you for your attention.